An Introduction to Taxonomy. Many centuries ago, in fact about 2,500 years ago, there was a natural history scientist by the name of Aristotle. He really didn't think of himself as a scientist at the time, but more of a philosopher, and he thought an awful lot about things around him. He actually began the foundation of the classification system that we use today, giving organisms two names to describe what they looked like, where they grew, trying to find relatedness among different organisms. It was a very broad classification scheme. Today we use a more specific type of scientific nomenclature or naming system, but Aristotle started it many hundreds of years ago. The person that's recognized now for the two-name or binomial nomenclature system that we use today is Carolus Linnaeus. In the mid-1700s, he was a up-and-coming physician and scientist, and scientists and physicians at the time had to also have degrees in botany because, of course, that's where we found most, if not many, different kinds of treatments for diseases in the plants, in the forest, and in the fields at the time. He determined to seek out the greater diversity of nature and organize or arrange them based on certain kinds of scientific names for the greater glory of God. He was a very scholarly person, very devout, and really is the founder of modern taxonomy with the binomial nomenclature system that he perfected or improved upon based on one that Aristotle had started a good 1,700 years before. Ultimately, whether we use Aristotle's system or the modern system that Linnaeus gave us, we're really trying to determine what kind of individuals are unique and have very specific criteria that we call the species. The species are essentially going to only be able to produce offspring by themselves or with their own kind. They do not interbreed with other species to produce either viable offspring or offspring that are still capable of reproducing. We say that species breed true, or that if we have one generation, that the species will give rise to identical or very, very similar offspring. An exception would be something like a hybrid. Hybrids are non-reproducing offspring from two different species. An example that's very traditionally used is the mule. A mule is an offspring if you breed a horse and a donkey. Donkeys are considered to be unique species, separate and different from horses. You can clearly see that they look quite different, even though there are some similarities. Well, if you produce a mule is a non-reproducing or non-productive hybrid. It still is an organism. It's capable of doing some of the same kinds of activities that you would find in both a horse or a donkey, but it is a unique non-species example of a hybrid that we wouldn't consider a new species, or would not consider a new species. When we're grouping organisms, we're trying to determine phylogeny, P-H-Y-L-O-G-E-N-Y, phylogeny, or relatedness. We group organisms together based on similarities and differences. We give them names and try to figure out how these different groups of different species are essentially related to one another, if in fact they are. So how do we group them together? Well, that would be the Scientific study of systematics and classification would be putting them into groups. Taxonomy, giving them names. All are very active areas of scientific research. Now, using the scientific names and trying to organize groups of organisms based on similarities and differences, scientists have come up with several different schemes or ways to group and classify organisms. Now, we've mentioned the binomial or the two-name system that originally started with Aristotle and that Linnaeus perfected and expanded upon. The two names, genus and species, are the foundation that we use for the unique individuals that have very specific characteristics. The word genus is always capitalized. The species name or the species epithet is not capitalized. If we're writing this to describe a unique species, we need to italicize both words. And you can see on the table on the slide some key examples of how names for different organisms include some types of descriptors, either the name of the individual who discovered it or some kind of growth characteristic of the particular organism that's unique to that organism. Now, organizing the different species and genera, which is the plural for genus, into sort of common groups. If we look for fewer and fewer distinctive criteria, in other words, we're looking for some very general common characteristics, then we can start to organize or group the different species within different genera, and then working our way up to the top of these little triangles where we have kingdoms with very few specific characteristics. We find out that many of our organisms can be grouped together into larger and larger groups. Now, there are various ways to figure out what group our organisms will go into. These are called taxonomic keys. Taxonomic keys can be arranged and devised in a variety of different ways. Regardless of how they are organized, we're still trying to find common similarities and differences between the different genera and species. The five-kingdom system has been used for quite a few decades to group organisms with basic, common, similar characteristics. Unfortunately, the kingdom, Monera, and Protista are no longer useful groupings. The organisms are too diverse, or the criteria that were used to classify organisms, let's say in the kingdom Monera, really aren't well defined, not good ones to use. Animalia and Plantae are still commonly used as groups for unique organisms because the criteria to classify an organism in either Animalia or Plantae are pretty straightforward. Animalia, we find eukaryotic celled organisms that have tissues and organs, and they are multicellular, and they are not photosynthetic or non-photosynthetic. So, eukaryotic, multicellular, tissues and organs, non-photosynthetic. It's an animal. Plantae, eukaryotic cell in structure and organization, multicellular, tissues and organs can be found, but we do have photosynthesis used as a metabolic process. So it's pretty straightforward to figure out if something is a plant or an animal. The kingdom fungi has been a useful one. Here we have organisms that are eukaryotic in cell structure, multicellular, but they do not have organs or organized tissues, and they are non-photosynthetic. So the fungi clearly have some characteristics similar to animals, some characteristics similar to plants, but because they do not have organized organs, they wind up in a kingdom all to their own. This table on this slide gives you even greater detail about differences within the 5 different kingdoms and when comparing between the 5 different kingdoms. Notice that the kingdom Monera have all the prokaryotic celled organisms, and then the other 4 kingdoms, Protista, Fungi, Plantae, and Animalia, have eukaryotic celled organisms. Then if you look at cell organization and other kinds of requirements, you can further define or detail which kingdom an organism might be classified in. Here we have the kingdom Monera. Again, not as contemporary as could be, but is still referenced today, where we find all prokaryotes. That would include the cyanobacteria, the archaea, and the bacteria. A scientist by the name of Berge, many decades ago, put together a key or a reference manual, which was Berge's Manual of Determinate Identification for Bacteria. He essentially created a way for scientists to determine and distinguish different kinds of species of bacteria that had been discovered. It's been updated, and we continue to use Berge's Manual, which is quite famous, for the distinguishing of and identification of bacteria and now archaea. Some of the criteria for distinguishing and classifying different types of bacteria are ones that we will use in the laboratory this semester. We'll take a look at the presence of the cell wall. What kind of cell wall composition does it have? How does it look when we stain it with different kinds of stains? What kinds of morphologies do we have? Are they spherical? Are they rod-shaped? We will perform biochemical tests to distinguish different species of bacteria that we'll work with in the laboratory. In addition to those first 4 different criteria that we'll use in lab this semester, there are other types of methods that we can use to distinguish different types of bacteria. Serology and phage typing. In addition to the previous list of criteria that could be used, we can also use amino acid sequencing, looking at the biochemical profile of both the fatty acids and DNA. We can look at the ribosomal sequencing of a particular component called an RNA subunit. We can perform polymerase chain reaction and other kinds of distinctive characteristics. Of all of these items on our list of criteria for classifying bacteria, the one that's currently used as sort of the ultimate determination of unique species is the sequence of the RNA subunit of the ribosome called the 16S RNA subunit. There are specific institutions and agencies all over the world that's what they do. We send them samples of what we think is a new species of their 16S RNA subunit, which is a unique little piece of a structure that we call a ribosome, and then those agencies help to determine, is this a unique structure? Do we really have a true new species of discovery? Keep in mind, even archaea that are prokaryotic cells will be classified based on certain biochemical differences and genetic differences. The kingdom Protista includes lots of eukaryotes that we find all over the world, especially out in the ocean or in aquatic environments. They're so diverse in nature and function. This is one of the kingdoms that's been a very hot area of research because of the great diversity. Like the kingdom Protista, organisms classified in the kingdom Fungi have eukaryotic cell structure, but the big difference, in the kingdom Fungi we find both single-celled and multicellular organisms. They are non-photosynthetic, and they have quite distinctive characteristics different from the protists. As I mentioned earlier, if we're going to classify an organism as an animal, it has to have eukaryotic cell structure, it has to be multicellular, with distinctive organs and tissues, and be non-photosynthetic. That includes all kinds of vertebrates and invertebrates within this kingdom. Now, an observation looking at the relatedness of different organisms and also looking at archaeological evidence, geological evidence, and comparative anatomy. Scientists have developed another theory in biology called the theory of evolution. Evolution is a mechanism that can be observed in organisms. We can even set up little experiments to design and demonstrate that the process of evolution is simply a mechanism for adaptation to an environment. Several theories that we've discussed so far, in addition to the cell theory of biology, we've talked about the theory of biogenesis. One cell gives rise to another. The germ theory of biology, where diseases or infectious diseases are caused by pathogens or disease-causing microorganisms. And now the theory of evolution, the fact that organisms adapt and react to their environment, and we can see and determine relatedness based on some of these characteristics of growth and adaptation. When scientists try to map out the essentially common ancestors, or try to map out the relatedness and essentially the genealogy of organisms on Earth, we look at the similarities and differences in our cell structure. And originally scientists thought that the relatedness or the tree of life was exactly that, just a tree, with lots and lots of separate branches. What we find now is that it's not really a tree, but we have quite a bit of a shrubby or sort of a very hectic looking family of origin. As scientists worked out the details of origin of cells and relatedness of one organism to another, it became pretty apparent that actual fundamental cell structure and organization seemed to be very important. And if we took a look at the fossil evidence, the biochemical similarities and differences, we found that the origin of cells was an important piece of information to determine. And so scientists have been working diligently for quite a few decades to determine when organisms and when cells started to appear on the surface of the Earth, what kind of evidence or data or things that we can measure, which as scientists, that's what we want to look for, and that's what we determine and make our decisions on. We use all of our senses, well, maybe not the sense of taste, but we often use sight, sound. We have to have something we can measure, that we can actually analyze, and touch, see, and feel to put into our list of information and criteria to figure out, where did these cells come from? How old are they? We use a lot of geology to help us to figure out where and when these things occurred. And so the origin of cell and the process of evolution, which is a mechanism that cells use to adapt and get sort of advantages in different environments, have been important criteria to use to figure out the origin and the relatedness of cells. In this chapter in this textbook, the author goes to quite an extent describing essentially the interrelatedness of organisms and the fact that we really don't have a tree of life, that more accurately, if we wanted to describe where cells came from, where different kinds of organisms probably emerged, is essentially a shrub of life. We have quite a bit of genetic exchange or exchange of genetic material among the different kinds of organisms over the billions of years it took for cells to evolve on Earth. we find that prokaryotes started to emerge on the surface of the Earth about 3.5 billion years ago. Roughly 1.7 to 2 billion years ago, eukaryotic cells started to appear. And if we look at the biochemistry of these different types of cells and the different groups of organisms within each sort of cluster of different kinds of cells, we can determine, again biochemically, just how long ago were these blended together or coming from the same common ancestor. The phrase lateral gene transfer is one that we will come back to when we talk about microbial genetics. In terms of taxonomy, we're simply looking at how similar or different the genetic material is amongst different cells, and the fact that particularly prokaryotes are quite prone to share genetic material amongst themselves quite freely. We find that the exchange of genetic material, sort of the mixing and matching of hereditary material from one organism to another, often will give an organism some kind of unique advantage, or maybe disadvantage. But it does lend to a sort of fluid motion or movement in the evolution or the adaptation of organisms here on Earth, from one generation to the next.